Dear friends and colleagues, good morning, good afternoon, depending on where you are. My name is Brian Chung. I'm the immediate past president of the Asia Pacific Society of Human Genetics and also the chief medical and scientific officer of the Hong Kong Genome Institute. I would like to welcome you all to the eighth joint Distinguished Scholar Series. As we all agree, sharing knowledge and experience is the key to accelerate the development of genomic medicine. And Hong Kong Genome Institute is committed to fostering collaborations and exchange of insights. With such a shared goal, we actually partnered with the Asia Pacific Society of Human Genetics to co-host this Hong Kong GI-APSHG Distinguished Scholar Series, which brings together renowned clinicians, scientists, and researchers to facilitate international knowledge exchange. And today, we're very happy to have Professor Jun Yang giving us a talk on pharmacogenomics. Let me do a brief introduction of Professor Yang. Professor Yang is the professor and endowed chair in pharmacogenomics at St. Jude Children's Research Hospital, and he is a founding member of the Pharmacogenomics Research Network. He completed his PhD training at Purdue University, Indiana, and moved to St. Jude, where he transitioned from postdoc training to faculty in 2010. Professor Yang currently serves as the Vice Chair for the Department of Pharmacy and Pharmaceutical Sciences. Professor Yang's research focuses on patient-to-patient variability in cancer treatment responses. He is committed to genomic and pharmacogenomic research of pediatric leukemia and leverages multidisciplinary collaborations to ask scientific questions on a global scale. His work primarily focuses on pharmacogenomics of leukemia treatment resistance and how genomics can give insights into anti-leukemia drug sensitivities and resistance. He has authored a number of papers on germline genetic mutations, racial and ethnic disparities, and how genes variants generate susceptibility to cancer and to treatment failure. Without further ado, Professor Yang, thank you very much for giving the lecture.
Thank you, Brian, very much for the kind introduction and also Wei for inviting me to this very distinguished seminar series. So what I was asked to do was to talk about pharmacogenetics and pharmacogenomics, part of it as an educational seminar, but also talk about some of our research work to stimulate some discussion around the discovery and clinical implementation of pharmacogenetics in the context of precision medicine. So this is the outline of my talk today. I will start by introducing the concept of a intra-patient variability in drug trace and genetics. And I will go over some basic concepts on pharmacogenetics, a brief history, candidate gene versus genome-wide approaches to do pharmacogenetic discovery. And I will also talk about specific use case, focusing on some of the research that took place in my own laboratory here at St. Jude's, specifically for pharmacogenomics of therapeutic toxicity. And the last part of my talk, I will give some examples of how to implement pharmacogenetics in clinical practice. So as a pharmacologist, we all know there is a generally positive correlation between drug dose on the x-axis and drug response on the y-axis. So the more drug you give, you usually expect more response from that individual. However, there are individuals who deviate from this line of correlation. For example, this particular patient here at the bottom right corner. So he seems to be very resistant to this drug because even at a very high drug dose, the response was minimal. By contrast, this individual on the top left corner is the opposite pattern. So even at a very low drug dose, she experienced lots of toxicity. So this is what we consider as an extreme response. So pharmacogenetics fundamentally is about interpatient variability. The goal of pharmacogenetics is to identify the source of intrapatient variability and we want to use this knowledge to guide pharmacotherapy. End of the day, it's all about giving the right drug to the right patients. So why do we care about genetics? I think in a way we're asking the question, to what extent does genetic variability contribute to the differences in the phenotypes we care about? In this particular case is either drug response or drug efficacy, or sometimes pharmacokinetics as well. So this cartoon that I took from New York Times actually illustrate this point very elegantly. So I think it's always a combination of nature versus nurture. Nurture being the environmental exposure factors that they're not inherited from your parents. But nature will be the things that you is actually in your germline DNA, which is drive the genetic contribution to these phenotypes. So now let's talk a little bit about human genetic diversity. I think I can probably go through this very quickly, as many of you are familiar with this concept being in the Hong Kong Institute of Human Genetics. We know that almost all human genes are polymorphic, single nucleotide polymorphisms, as illustrated here. This is the most common variation. There are about 4 to 5 million SNPs per person. 99.9, essentially all genes have at least one coding variants recorded in various different databases or cohorts. And 50 to 60% of the coding variants are missense, meaning they actually change the amino acids and potentially affect the function of the protein the gene encodes. And then 99% of the genes actually have at least one nonsense SNP, which in a way is very alarming because almost most genes have been truncated in the germline at least once in the human population. So this is the older data sets from whole exome sequencing data that's published, almost 17 years ago. In thousands of patients, they show that on average per person has 46 nonsense coding variants, and also per person there are 52 frameshift indel variants. So altogether, about 100 genes per person carry substantial germline defects. As I said, this is actually a pretty substantial number, thinking about people walking around with truncating mutations in their germline genome. So if genetic variability is so ubiquitous, what proportion of the variability in response to mutation is actually related to genetics? So this is the old data looking at pharmacological traits, in particular pharmacokinetics of some of a few medications, and this is actually published in Goodman-Gilman. For those of you who are pharmacologists, this is a classic textbook. They're looking at clearance of specific medication in identical twins versus the fraternal twins. So the black bar is one of the two twins, and then the gray bar is the other of the twin pair. So as you can see, if it's identical twin, the drug clearance is almost completely the same across two individuals from the same twin pair. But if it's a fraternal twin, there's actually quite a bit of difference in terms of their pharmacokinetics or clearance of this drug. So it was estimated for pharmacokinetic traits, something between 75 to 85% of the inter-patient variability is actually inheritable. So what about pharmacodynamics? What about the effects of the drug to human body? And this is an experiment that was also done many years ago, almost 22 years ago, by Eileen Dolan's group at University of Chicago. What they did was actually a very clever experiment. They took cell line, EBV transformer lymphoblastoma cell line from the Thousand Genomes Project. You can see these are cell lines prepared from large categories, and they identified family child trios, like the blue, red, and green here. So these three individuals is considered as trio. What they did was to measure the cytotoxicity of daunorubicin for these three individuals, both parents and child. As you can see, one of the parents seemed to be quite resistant to daunorubicin. So this is the IC50 curve. survival on the y-axis and concentration on the x-axis. And then the other parent seems to be very sensitive. And then the child is somewhere in the middle, which again aligns with our understanding of how genetics works. So pharmacodynamics, the effects of drugs to human body is also heritable, but to a lesser degree. Only something between 30 to 70% of pharmacodynamic traits can be explained by inherited genetics. So, like I said, pharmacogenetics is all about linking genetic variability to variability in drug response. So, the goal of pharmacogenetics is to evaluate how an individual's genetic makeup corresponds to the response to a specific medication. And at the end of the day, we want to use this information to inform individualized therapy to increase effectiveness while reducing side effects. So pharmacogenetics. Traditionally, we think about at least two classes of genes that are important for pharmacogenetics. The genes that encode enzymes responsible for the metabolism or disposition of a drug. So this could be enzymes, the P450 enzymes in the liver, could be drug transporters, the ABC transporters, the SLC transporters that exist across different organs and tissues. And genetic polymorphisms can also exist in genes that encode drug targets. This could be receptors that are inhibited by the drug or enzymes inhibited by the drug and cancer targets as well. So genetic variants in any of these genes can potentially influence both the pharmacokinetics and also pharmacodynamics of specific drugs. So now let's talk a little bit about the history of pharmacogenetics. In a way, we think about pharmacogenetics, the evolution of this field, maybe in three different phases or eras. In the back in the 1950s, this is what I consider as an idiosyncratic era. So people started observing differences in response to modern medicine. For example, in 1957, it was famously argued that some idiosyncratic drug reactions are likely produced by specific genetic mechanisms. That actually provides the conceptual framework. But at that time, of course, no DNA sequencing, they didn't identify specific genetic material or components. 57, there was a nice paper in JAMA talking about some specific examples, especially G6PD deficiency as a biological basis for hemolytic anemia, especially from anti-malaria drugs. So 59, the term pharmacogenetics was officially coined. It was published. I described it here. But again, this is the idiosyncratic. We don't really know what pharmacogenetics, or at least we don't know the genetic causes, no, not the gene, not the variants. It just conceptually thinking this is something related to genetics. So in the 60s and 70s, this is what I consider as the biochemistry era. And at this point, scientists start isolating enzymes. They start doing this in sort of the families. The 1960, the Price of Evan actually studied the metabolism of isoniazid, right? In families, and they demonstrate that this is actually a Mendelian trait, meaning it actually follows the rule of genetics. In 77, it was additional studies looking at metabolism and the deprecoxyquine. And 79, Eckerbong and others looking at sparating. Again, these are studies illustrating the polymorphic nature of drug metabolism. No genetics whatsoever in the 60s and 70s. It's all about biochemistry, isolating the enzyme, looking at enzymatic function, especially when it comes to drug metabolism. So in the 80s, 90s, I think we started entering the molecular era. 1984, CYP2C19, a very important P450 enzyme, was identified. 87, CYP2D6, was molecularly cloned. In 89, NAT2 was also cloned. This is the enzyme responsible for the metabolism of isonucleotide. 93, C2C19 gene was cloned. So with the advances in molecular biology, people start realizing the genes that are responsible for producing the enzymes that are related to the metabolism of drugs. So if we think that 30 to 90% of the variability in drug response, either pharmacokinetics or pharmacodynamics, is related to genetics, but how do we pinpoint the specific factors or genes or variants in a gene driving drug response and toxicity? I think generally speaking, there are two different approaches. The first one on the left is what we consider as candidate gene approach. So in this case, we assume we know how the drug works, and therefore we only focus on genes related to this specific drug. We know which enzymes are important for the metabolism of this drug. We know the target of this drug. So we're just checking polymorphism in the genes we think are relevant. Another approach, which is agnostic, is a genome-wide association or genome-wide scan. Here, we do not assume that we know everything about the drug, and therefore we actually want to look at all genes in the genome and all variants, or as many variants as possible, to look for the genetic factors driving the drug phenotypes of interest. So two different approaches complementary to each other. And now I'm going to walk you through a specific use case and to illustrate the value of both candidate gene approach and also genome-wide approach. So before I talk about the drug, let me first introduce the disease for which the drug is used, and also the disease that I spend most of my time thinking about. So it's a cancer, acute lymphoblastic leukemia. It's a specific type of blood cancer. It's characterized by this uncontrolled proliferation of undifferentiated blasts. You can see that under a microscope. In normal bone marrow, you have a heterogeneous group of immune cells, B cells, T cells, neutrophil, macrophage, etc. But in the bone marrow of a leukemia patient, you have this very uniform composition, which is 99% of a leukemia blast, because all these undifferentiated cells, they're now proliferating very fast. They just crowd out all the other cells. And this cancer is particularly interesting to us because it's the most common type of cancer in children. So in the US, there's about 3,500 new cases every year. About half of them are in kids, which is highly unusual because when we think about cancer, we almost always assume this is an adult disease. It's very, very rare for a cancer to have more than 50% of the patients under the age of 18, and that's certainly true for ALL. Another interesting thing about ALL is actually one of the first cancer that were subjected to chemotherapeutic agent therapy or modern medicine, so to speak. So this is a list of drugs that we currently use for ALL treatments, and most of which are very, very old. The first drug, mercaptopurine, was introduced in 1953. Methotrexate the same year. Prednisolone, dexamethasone, 2-glucocorticoids, 58, 55. And the vast majority of the drug, as I said, are at least 60 or maybe 70 years old. So over the past 70-something years, we barely introduced any new drug. It's always the old cytotoxic chemo drugs. However, they work remarkably well. So what I'm going to do is to focus on purine and tell you about why pharmacogenomics is particularly important for this drug. So purines, there are two of them. These are chemical analogs of the naturally occurring guanine, which is ATCG, that's the G. So the only difference between thioguanine and guanine is that now you have a sulfur thiol group on the top. So there's a single modification make this a drug instead of a naturally occurring base. And this drug is very, very important for acute lymphoblastic leukemia, but it's actually used even more commonly in patients with inflammatory bowel diseases, and which is a much bigger population in the US, and certainly that is also true in Hong Kong. So for ALL, this is a very important therapy. Children and adults with this cancer will have to take this drug daily for about 1.5 years up to 2 years every single day. And myelosuppression is the most common side effect of valproate. Your white blood cell count will drop when you're on this drug. And dose titration is actually pretty difficult for this drug. It's based on WBC and C, because if you have myelosuppression and you back off on valproate therapy too much, you actually subject patients to less intense therapy, and you put them at higher risk of relapse because their leukemia cells will not be under proper control. But if you do not dose reduce sufficiently, you expose them to excessive toxicity, which can also cause treatment disruption, and you stop chemotherapy altogether. Of course, that will also lead to risk of relapse. So this is a very fine balance you have to strike between toxicity and efficacy. And this is also why pharmacogenetics is so important for cancer therapy, because the therapeutic index is always so small. When the therapeutic index is huge, you don't really care about individualization because nobody gets toxicity. I mean, nobody gets toxicity from aspirin, and nobody will want to do a pharmacogenetics study of aspirin. But then for drugs like valproate, because it's so toxic, but also so effective, you really have to think about how to individualize and be very precise about it. So this is a prodrug. Mercaptopurine structure is showing on the left. And being a prodrug, it actually requires many steps. Enzymatic reactions intracellularly to be activated. First, it needs to be ribosonated, adding the ribose, and then this nucleoside needs to be phosphorated, monophosphate, diphosphate, and triphosphate. This triphosphate, TGTP, structurally it looks very similar to GTP, and it actually gets incorporated into DNA double strand very easily because chemically it looks just like GTP. Cells will not be able to recognize the difference. And once it's inside the DNA double strand, it's not a perfect match. Usually we have G2C match with the hydrogen bonding, but because you have a para group, the match is not perfect. So the DNA looks like there's a bubble. And the cell will recognize this bubble as a mistake. You try to cut it out and try to fix it. But usually they cannot. And after a few rounds of repairing, you will just the cells will just say, well, I can't fix it. I'm just gonna die. So that's the mechanism of action. So you have DNA incorporation of the chemically modified guanine, and then that eventually leads to DNA damage and cytotoxicity. So back in the 80s, an enzyme was identified called TPMT, valproate methyltransferase. The only known biological function of this enzyme is to add a methyl group to mercaptopurine. And this simple reaction will make mercaptopurine completely inactive, right, as the methyl group is not active. So now the therapeutic efficacy of this drug is the battle between the activation pathway going from left to right, and the inactivation pathway going from top to bottom. In the early 90s, several investigators in the U.S., Bill Evans Barry at St. Jude and Dick Weishaborn at Mayo Clinic and others, started sequencing the TPMT gene to look for genetic polymorphism. And they identified 3 of them. This one here, G238C, and then G468 and 719G. So all 3 variants turn out to be deleterious, meaning they actually lead to the reduced function of this enzyme. And why is this important clinically? Because in 1999, Mary Riley and Bill Evans published the landmark paper in JNCI, describing that the individuals homozygous for the CC allele, which is a defective allele, they have no detectable level of TPMT enzymatic activity, and they cannot inactivate the drug. So every single molecule of 6MP is activated as a TGN. Now you have this excessive amount of active metabolite. You have 100% risk of myelosuppression toxicity. If you're heterozygous, then your risk of toxicity is only about 35%. If you're wild type, it's only 7%. So this is remarkable because now you can actually use the genotype to predict who's going to have toxicity from this drug. So based on that, now you have two ways of giving this drug. The conventional way is to give everybody the same dose, regardless of the genotype. You don't even bother genotyping or sequencing them, and just let them crash and burn because some of them cannot tolerate. So these patients will have a lot of the active metabolite, a very high risk of toxicity. But you can also be smart about this, genotyping everybody up front, and use the genotype to guide your valproate dosing. If you're homozygous, dose reduced by 90%. If you're heterozygous, dose reduced by 50%. If you're wild type, 100%. And that essentially normalizes the exposure to the active metabolite and mitigate or completely reduce the excessive toxicity as that you expect to see in patients with heterozygous or homozygous for this defective allele. However, even patients with GG phenotype, wild type, still 7% of them experience toxicity. So there must be some other genes contributing to this toxicity. So Belinda is a patient who actually offered us a window to look into this. She was 5-year-old when she was diagnosed with acute lymphoblastic leukemia. She was ethnic Chinese in Malaysia. Went to Singapore, National University of Singapore Hospital for her leukemia therapy under the care of Professor Alan Go, who's a very good friend of mine, also a collaborator. So what's remarkable about Belinda is not her leukemia, it's actually her toxicity to valproate. So on the top, you can see this dose titrating curve, the green curve right here, describing the dose fluctuation for 6-month capture variant or 6MP over the course of 71 weeks. So for average patient, it goes up and down, but always hover around 50 milligram per meter squared. That's the standard expected dose for Asian kids. But for Belinda, it was very, very different. She actually started pretty low, and within 3 weeks, her WBC was completely wiped out by valproate or 6MP. She has so much infection because you have no WBC. She was admitted to the ICU for severe infection, and to the point that Alan had to stop her chemotherapy, or particular 6MP, for her counts to recover. But you cannot cure a child with leukemia without 6MP. So he actually put her back on 6MP, and when he did that, her WBC just plummeted again back to 0. A lot of infection, ICU admission, and then wait until it can recover. You give 6MP again. It just happened just like last time. So this went on for several months. Eventually, her stable dose was 2.8 milligram per meter squared per day. 2.8 milligram per day is drastically lower than the standard dose, which is 50 milligram per meter per day. And the first thing Alan did was to genotype her for TPMT because we know this is the important pharmacogenetic gene and variant. And she turned out to be wild type. So now we think we'll have to do something else to figure out. So the candidate gene approach didn't work. We decided to take a genome-wide associations approach. So the way it works is you identify 2 groups of individuals, those treated with this drug, experiencing toxicity, and then your controls will be individuals treated with the same drug but did not experience toxicity. And you got the germline DNA, you genotype using SNP array or whole genome sequencing, and then you compare the frequency of these variants or each variant between cases and controls. And at the end of the day, you plot the data in the format of MATLAB, which I'll talk about next slide. So we decided to do a genome-wide association study to look at genetic variants comprehensively for their contribution to 6MP toxicity. So we collaborated with Doctors Mary Relling and Smita Bhatia, taking advantage of the Children's Oncology clinical trial AAL03-1, about 700 patients, all treated with Puring for ALL, a replication cohort of a little bit less than 400 patients here treated at St. Jude's. So we use dose intensity as a proxy marker for toxicity, genotype about 250,000 variants, mostly coding for this cohort. So this is our math and plot, the GWAS result. So each dot here is a SNP going from chromosome 1 to the 6th chromosome. And the y-axis looks transform the p-value for the association between genotype and 6MP toxicity. So as you can see, there are only 2 dots or peak that that went above the genome significance threshold 5 times 10 to the 8. The first one is in TPMT gene on chromosome 6, which is our positive control. It works beautifully. And then there's a new gene on chromosome 13 called NUDT15, which we didn't know much about at that time. So this is just to show you an association between the NUDT15 genotype and 6MP dose intensity. You see, if you're homozygous, you have this drastic toxicity and dramatic dose reduction, only not like 8% of standard dose. If you're heterozygous, it's somewhere in the middle. There's also treatment replication cohorts. And actually, this is for TPMT. Let's skip to the NUDT15. Here we go. Yeah, 92% dose reduction and heterozygous in the middle. And this index variant from GWAS is the actual coding variant in exon 3 of NUDT15, causing an arginine to cysteine change. I don't want to go into details because the paper is already published by us in 2016, describing the biochemistry. So it turns out NUDT15 is a pyrophosphatase. It actually cuts off the 2-phosphate from the triphosphate form of this compound, TGTP, converting that to TGMP. Because there's only one phosphate, this molecule here or metabolite cannot be utilized by DNA polymerase to get into DNA. So NUDT15 is actually an inactivation pathway or reaction for Pharmacogenomics. And the variant that we identified turned out to be deleterious, causing loss of NUDT15 enzymatic activity. So now if you have the genetic variants, you have much lower activity of NUDT15. You cannot complete this inactivation process. You end up with a lot of TGTP, the active metabolites. So you have a lot of incorporation of this chemically modified guanine. And that leads to excessive DNA damage and toxicity in patients. This is the sort of molecular mechanism linking NUDT15 to therapeutic toxicity. So I think the goal of pharmacogenetic research are almost always twofold, at least. We try to have a better understanding of the mechanism of adverse effects or efficacy. We want to use that information to inform drug interactions, drug dosing, and timing. And I would say 99% of the time when we do pharmacogenetic discovery research, we don't always identify something clinically actionable. It's just a remote possibility that we will discover something like NUDT15 with a very large effect size. NUDT15 variant actually has a odds ratio of 32 in terms of predicting therapeutic toxicity. And we have an odds ratio of that magnitude. Of course, that can enable clinical genetic testing, which can be used to guide prescribing. So now let's talk about pharmacogenetic implementation in the clinic. So actionable pharmacogenetic gene drug pairs have been established for a very long time. I talked about at the beginning of my presentation. Back in the 50s, it was recognized that G6PD deficiency is linked to hemolytic anemia secondary to primacrine treatments. And 88, 89, 96, 2002, there are all sorts of drug gene pairs. CYP2D6 in the coding, valprorin and TPMT, EPYD for the excessive myelosuppression from 5FU, HLA genotype for association with Stevens-Johnson syndrome linked to a back review. And there are many more after that. However, the clinical implementation of pharmacogenetics is much, much more slowly, despite the overwhelming evidence, either clinically or scientifically. So, for example, this study published in 2017, looking at over 132,000 individuals, covering over 3 million hospital clinical visits in the U.S. There are something like 268,000 medication orders for 95 drugs, which actually contain pharmacogenetic testing, specifically in their FDA-approved drug label. So you will consider these drugs are great candidates for clinical implementation. So 49 of these 90 drugs are actually deemed clinically actionable by CPIC, which I'll talk about in a minute. However, looking at all these medication orders, only less than 1% of them were accompanied by testing in these healthcare systems in the US. It's only 1.5, even when the FDA drug label specifically says recommended or required pharmacogenetic testing. So the uptake of pharmacogenetic is very poor, at least back in 2017. And then the survey was performed by many of us in pharmacogenetics or PGRN to ask in practitioners, whether physicians or pharmacists, to identify the top 3 challenges to implementing pharmacogenetics in clinical practice. And 95% of respondents selected process required to translate genetic information into clinical action. It's not about genotyping patients. That's the easy part. But once you have the genetic information, how do you actually use that to guide medication or pharmacotherapy? That's the most challenging part for most people in the field. So it was then dawned upon us that actually it's very important to develop clinical guidelines to teach people, or to at least to inform people in a very structured way how to translate the genetic information into prescription. So driven by that, the Clinical Pharmacogenetics Implementation Consortium was formed in 2013. And what it does is to curate, and post freely available peer-reviewed evidence-based and updatable detailed gene drug clinical practice guidelines to specifically tell people how to go from genotype to prescription, individualized prescription. So the guidelines on specific clinical actions for patients with genotype. So for example, if I'm homozygous for NUTT15 star 3, what dose of thiopurine should I get? So this is what CPIC guidelines are all about, translating genetics into prescription. So CPIC is actually a global organization with more than 780 members in 590 institutions covering 52 countries. The guideline authors are very international. 23% are actually outside of the US. Almost every single continent, of course, nobody from Antarctica. So a very important part of the CPIC is to evaluate or vet the evidence for clinical implementation. It goes through this very structured and sophisticated process to evaluate every gene-drug interaction or association, to classify them as CPIC level A or B, which are clinically actionable, and then CPIC level C and D. Those are informative, but not warrant clinical action. So I think TPMT, IUNTT15, these are very important for captive purine, clinically actionable, but then CYP2D6 and propionyl, they're real association, but not strong enough for clinical action. The CPIC guidelines are posted on their website. It's freely available. There are no limits on how you want to use it. It's peer reviewed and published in Clinical Pharmacology and Therapeutics, which is a journal for American Society of Clinical Pharmacology and Therapeutics. It's a very standardized form across different drugs, the different genes. It's fairly easy to use. The grading of the evidence and recommendations are all transparent. The information and process are all posted. It can be updated on the website ahead of a publication. If there's a new discovery about specific variants that warrant consideration, CPIC will add that prior to the actual publication. And it's called the entire process really modeled after the structure put in place by the Institute of Medicine, part of National Academy of U.S. National Academy of Medicine. So there are right now 28 guidelines with 17 updates covering 34 genes and 160 drugs as of 2025. So all of them are listed here. And how many drug gene pairs should be used in the clinic? So if you look at the prescription in the U.S., something like 80% of the prescriptions in the U.S. healthcare systems are affected by actionable pharmacogenetics. This is a very sizable proportion, right? Almost 1 in 5 prescriptions in hospital systems in the U.S. can be influenced by pharmacogenetics. And these are clinically actionable pharmacogenetic associations. And the listing of FDA labeling for drugs that's this provided on the PharmacoGKB website. You can also find that information in addition to the CPIC information. So have we done pharmacogenomics implementation here at St. Jude? Yes, we have. We have been doing that for many years, in fact. And we do that in the context of a clinical trial, which is called PG for kids. So the goal is to migrate pharmacogenetic testing from laboratory into routine patient care to be available preemptively through the electronic healthcare system. And we do have some exclusion criteria in terms of which ones may not be suitable for this clinical trial. So this is our process. We start with patient consent, then we collect the blood sample. We perform an array-based genotyping for almost 1,200 genes, and all the results go into a research database. And after that, the results are returned to prescriber as well as patients for education. And we do have a clinical decision support system, CDS, in our EHR. And right now, we put about 17 gene results in the EHR for clinical implementation. So these are the genes that we have implemented so far, going back to 2011, when we started with TPMT and the CYP2D6. UDT15 was added in 2017. So we discovered this in 2015, figured out the biology in 2016, and 2017 we implemented at St. Jude. It's a very, very fast-paced process for things to go from discovery to clinical implementation. So at St. Jude, looking at our patients, over 7,700 of them, 95% of them have at least one high-risk pharmacogenetic finding. You can see that 28% of them, 28% of them have 3 high-risk results, and 31% of them have 2 high-risk results or genotypes. So vast majority of the population will have some sort of a actionable pharmacogenetic finding, including those of you who are in the audience. And this is our active CDS, clinical decision support system. We have a pre-test situation. We have a post-test situation. And this is the window that pop up in our EHR. For example, with CYP2D6, we'll talk about how genotype is recommended before prescribing coding. And then this is our post-test alert, talking about the genotyping results and how that should be used in the event the drug is prescribed. So this actually enables us to roll this out in our EHR and has really facilitated clinical implementation of pharmacogenetics at St. Jude and many other hospitals and institutions in the U.S. have adopted similar systems, CDS, in EHR, including EPIC, as well as using CPIC guideline as the source information for their clinical implementation practice. So I hope that I have convinced you pharmacogenomics is a branch of genomic medicine, and the goal of which is to identify the genetic basis of intrapatient variation in drug response and toxicity. Most candidate gene studies only examine genes responsible for related to drug metabolism, transport, disposition, and they're believed to be directly involved in drug disposition. And genome-wide association studies, by contrast, look at all genes and all variants agnostically, and it can point to novel biology driving drug phenotypes, like I illustrated using NUDT15. CPIC, the Clinical Pharmacogenomic Implementation Consortium, has been leading the way to develop clinical guidelines for using pharmacogenomic for treatment individualization, many of which has been adopted really by regulatory agencies here at FDA in the US, EMA in Europe, and many other places as well. I think a lot of main groups have been trying to tailor CPIC guideline to their local practice, whether the different types of medication used, even if in the same class, or maybe the degree of dosing documents that they be modified as well. So it does take a village to do pharmacogenomics. A lot of work that I talked about are done by people in my lab in collaboration with colleagues around the world. As you can see, all the flags here. And of course, nothing can be done without money. So I have to thank my funding sources. In the end, I do want to show this one photo of Belinda. So she turned out to be homozygous for NUDT15 R139C. And she made through her chemotherapy, and she went back to school, and she is very healthy. I don't have that photo, but she is actually now in college in Australia, which is just so wonderful as a basic scientist to see that some of the work that we do can actually make a difference in patients and put a face to the genes and genetic variants that we study. And this is the acknowledgement for the implementation work that I talk about here at St. Jude's. I think with that, I'll stop here and be happy to take a few questions. Thank you very much for your attention.
Thank you very much, Professor Yang, for this very insightful lecture. I think you have shared a lot of interesting and successful story. And I'm most impressed by two. First, of course, is the story of Belinda. And now we all know how it leads to taking care of patient leads to important discovery of the role of NUDT15 in this important 6-MP drug metabolism. I still remember my days as intern. I have to work in the oncology day centers, and patient coming in for maintenance chemotherapy all have this drug level check. And many a times you see disappointed faces from the parents because the drug level is something that they need to titrate, and that prevents them from entering the next cycle of treatment. And of course, we all know that have a lot of undecided side effect, because if you do not continue the therapy, there is a chance that the cancers may relapse. And the other interesting story about implementation is about the story you told on CPIC, which is a database that we use quite often when we come into interpreting pharmacogenomics results. And thank you to you and the village who has contributed to CPIC, because that's making our lives so much easier with all those guidelines that you have provided us with how do we titrate the dosage when we know the genotype of those actionable pharmacogenomics genes. I'm going to pass the time to Dr. Ma to mediate the Q&A sessions. Dr. Ma, please.
Thank you so much. So we have one question in the chat box now, and you So this audience is asking about the PG4KDS. So has that project boost the use of PGX guidelines and by 100%? If not, what will be the future challenges and or hesitations for clinicians to follow such guidelines? So even after you implement that program.
Yeah, so we actually started that clinical protocol quite a few years ago, and I said we have 17 genes that are currently in our EHR that's been clinically available for physicians already. But we did do a study a few years ago to look at the uptake because we provide recommendations, right? You use this genotype, this is what those you will use. We did survey our oncologists and physicians here at St. Jude to look at how often they actually follow through to actually adopt recommendation. For things like 6MP, the adoption is almost 100% because this is a very essential drug for chemotherapy. You have to follow that very carefully. But another example is ondansetron and CYP2D6. So that drug, oftentimes you have to give them on the spot. You don't wait for genotype. So the adoption is not necessarily great. So I think there is some inherent difference across different medication, and that probably drives the difference in the uptake. So I think that's somewhat expected.
Yes. And the second, thank you for your answer. So the second question will be, nowadays, or most of these recommendations are based on single variants or gene panels. Do you see a clinical relevance to develop a polygenic risk scores for pharmacogenetics for a agnostic usage? For example, a risk score that predicts a general response for even for multi classes of drugs.
Yeah, that's actually a really good point. I think as this as he or she alluded to, vast majority of the clinically actionable pharmacogenetic associations are one gene, one drug, right? I think we have some rare examples. Warfarin, you have VCOC1 and CYP2C9, that's two genes. And thiopurine, you have TPMT and immunity 2 genes. But we never had anything more than two genes. So it's very straightforward to implement because at most you have two genes. And sometimes you might have more than two variants, 3, 4, or whatnot. Are we ready for polygenic risk score? I don't know. I feel in the disease biology space, there's a lot of hypes about polygenic risk score. A lot of that is because the how robust the disease GWAS has been, because you're talking about the genome association of type 2 diabetes, or maybe 100,000 patients or 300,000 controls. So you actually end up with very robust statistics for that type of genetic study, and you can reliably take the finding from those large GWAS to develop a polygenic risk score. My concern for pharmacogenetic is that our sample size is almost always very small, a few hundred, maybe a couple thousands. So if you have genome significant hits, that's great. That's probably reliable. So if you have something that is not genome significant, I really worry about the robustness of those hits. And if many of them are not real, then building a polygenic risk score using thousands of variants, that can be very problematic because the associations are not very robust, and you end up using the wrong variants to build polygenic risk score. So maybe one day we'll get there. But another point I want to make is now I have seen a lot of studies showing that polygenic risk score for disease biology, they are associated with the drug response or even toxicity. For example, there are polygenic risk score for liver function and baseline. And that polygenic risk score is actually associated with the liver toxicity as well. Meaning, if your liver is already pretty bad to start with, and then you give them a hepatotoxic drug, yes, they're going to have more likely to have liver toxicity. So I think in that specific context, maybe polygenic risk score makes sense, but it's not polygenic risk score specifically for the toxicity or drug response. It's actually polygenic risk score for biological trait that's linked to toxicity. I hope I answered that question.
Yes, and the third question will be, what do you think about the role of regulatory agencies? How could they do to support the pharmacogenetics in clinical practice?
Again, that's a really good question. It's interesting. I think it was August of last year in Singapore. There was a very famous or maybe infamous lawsuit. So was a patient was treated for some sort of skin condition at a private practice in Singapore and was prescribed azathioprine, which is one of the valprins. And the physician neglected to do pharmacogenetics, did not follow the myelosuppression very closely, and the patient actually ended being admitted to ICU because of the very severe myelosuppression, and he was in ICU for about 8 days. And then after that, he filed a lawsuit against the physician in Singapore, and the Singapore Health Authority actually handed down a suspension of license of that physician for I think couple of 1 or 2 years. And it actually generated quite a bit of interest in the lay audience as well as the medical community, thinking that there's actually significant consequence um, of not doing pharmacogenetics. You're doing harm to your patients, and there are legal consequences as well. So I think that's where regulatory agency can really step in and start looking at how to implement pharmacogenomics at scale to educate the practitioners to make sure that the things, the one that I just described, will not happen in the future.
Thank you. So I guess we can take another two questions. So the first one will be, have you incorporated AI into any part of your CPIC guide pharmacogenetics implementation? So if yes, like what components or which of the workflow does it support, and have you observed any benefits that AI can bring in clinical decision making?
So we actually have not, but I think that's a really good point. We have been thinking about it. I think the pharmacogenetic guidelines are pretty easy to implement. I think AI can be, it's probably very easy for AI to operationalize this, right? Because it's one gene, one drug, and we have a very specific instruction as to what to use. So I figure this will be again a low hanging fruit for AI assisted, but we haven't done that. But that's a very good point.
Okay, so thank you. So the last one will be, have you performed a microarray based GWAS study on the 657 TAL samples? Do you think that whole genome sequencing could identify additional candidate genes? Are there any drug gene pairs that are currently used in clinical testing for those TAL patients?
So, you mean the 2015 study, that was the GWAS studies. We actually genome type 250,000 and subsequent variants. Subsequently, we did a bigger GWAS testing, I think up to like 1 million variants. We didn't see anything more beyond NUD15 and TPMT. Those are the strongest. There might be other variants with smaller effect sizes, but they're not necessarily very reproducible or robust. There are many other drugs. There are many other gene drug pairs we're testing clinically for TLL. In terms of toxicity, it's really just thiopurine. That's the most common one. And we also do a respiricase and G6PD. That's another pair. But for efficacy, we do a lot of somatic sequencing, right? We're trying to identify mutations in for targeted therapeutics, but that's probably beyond the scope of the presentation today.
Thank you, Professor Yang. I know that the time is running short, and we probably need another hour of lecture from Professor Yang very soon to tell us more, not only about germline pharmacogenomic changes, but perhaps the somatic genomics changes important in cancers like childhood leukemia. But it's getting late, I think, in Professor Yang's side. So thank you very much, Professor Yang, for this exciting lecture. Please educate us more in the near future. Thank you very much, everyone, for attending. Goodbye for now.